Hello everyone, welcome. In this video we are going to discuss about amino acids and proteins. Now amino acids are the compounds that are made up of carbon, hydrogen, oxygen and nitrogen, and as you all know, amino acids are the building blocks of proteins. Now this amino acid is made up of amino group, carboxyl group, hydrogen atom and a side chain, and these all is bonded to an alpha carbon. Okay, that is how amino acid exists. Okay, it has amino group, carboxyl group, hydrogen atom and a side chain, all of this bounded to an alpha carbon. Now in alpha amino acid, the amino group and a carboxylate group, both of them are attached to a same carbon which is known as alpha carbon. Okay, alpha amino acid, there is alpha carbon, alpha carbon, amino group and carboxylate group are attached. That is how the alpha amino acid exists. Now, the different alpha amino acid have different side chains in them. Okay, alpha amino acid differs in side chain that is being attached to this alpha carbon, and the side chain is also known as R group. Okay, so in different alpha amino acids you will be finding different side chains or different R groups. Okay, side chain and R group both are the same thing. So this is how the general structure of amino acid looks like. There is an alpha carbon in the center, then to that hydrogen atom is attached and carboxyl group is attached, and then alpha amino group is also attached, and it has a side chain as well. So this is the general structure of your amino acid. Now that structure of amino acid that we just saw is common for all alpha amino acid except proline. Proline don't have that structure. Except proline, all the alpha amino acid have that structure that we just saw. Now this R group or side chain that has been attached to the alpha carbon is different in each amino acid. Okay, different amino acid have different side chains. Now in the simplest possible case, that R group is an hydrogen atom, and that amino acid in which the side chain is just a hydrogen atom is glycine. Okay, glycine is that amino acid who has hydrogen atom as the side chain or R group. So this is how the structure of glycine and lysine looks like. As you can see, the side chain of the glycine is just an hydrogen atom. Okay, R group is just an hydrogen atom in case of glycine, and this is the structure of lysine, and this whole structure is its R group or side chain. And as you can see, this carbon is alpha carbon, but this carbon is beta carbon, this carbon is gamma carbon, this carbon is delta carbon, and this carbon is epsilon carbon. So that is how they are named. Okay, so this is the structure of lysine and this is the structure of glycine. And glycine is the simplest amino acid. I mean, you just have an hydrogen atom as the side chain. Now, as we discussed, in alpha amino acid, both the amino group as well as the carboxyl group are attached to the same carbon, and that carbon is alpha carbon. Now there are some naturally occurring amino acids which are not found in protein. The structures of this naturally occurring amino acid is different from alpha amino acid. And how it is different? Well, the amino group of this naturally occurring amino acid is bound to the carbon atom other than alpha carbon atom. Okay, the amino group is not bound to alpha carbon. It is bound to some other carbon atom, and that is how the structure of these naturally occurring amino acid is different from alpha amino acid. And the thing that should be noted here is that this kind of naturally occurring amino acid whose structure is different from that of alpha amino acid are not found in protein. So those amino acid can be your beta, gamma, delta, epsilon amino acid, and this will depend on the location of the carbon atom to which this amino group is attached. Okay, if the amino group is attached to the beta carbon atom, then that amino acid will be called as beta amino acid. If the amino group is attached to the delta carbon atom, for example, then that amino acid will be known as delta amino acid. Now, amino acids can act as acids and bases. Now, let's discuss how is that. Now, amino acid has an amine group, that is NH2, and a carboxylic acid group, that is COOH. And this amine group is basic in nature, as you all know. It acts as an proton acceptor, and the carboxylic group is acidic in nature, that is, it acts as a proton donor. Okay, so the amine group is basic, it is a proton acceptor, and the carboxylic group is acidic in nature and it acts as a proton donor. And that is the reason why the amino acids are amphoteric. Okay, these amphoteric molecules can donate proton and can accept proton. Okay, they act as an acid or a base, this amphoteric molecule, and the amino acids are amphoteric, that is, they can act as an acid or a base because they are able to donate a proton as well as they are able to accept a proton. Now, if in the system if there is high concentration of proton, that is, the pH is low, then the carboxyl group of the amino acid will accept a proton and will become uncharged, and that is the reason why the overall charge on the amino acid will be positive. Okay, so if at low pH the amino acids are positive. Now, at neutral pH, the amino acid in the aqueous solution exists as dipolar ion or a Zwitter ion. Okay, at neutral pH, amino acid is Zwitter ion. Okay, now what is this Zwitter ion? Well, Zwitter ion has no overall electric charge on it. Okay, it contains both positively charged group and negatively charged group, and both of these cancel each other, and that is the reason why Zwitter ion has no overall electric charge on it, and that is why this amino acid in aqueous solution exists as a Zwitter ion, because it has a positively charged group and a negatively charged group. So that is the reason why at neutral pH the amino acid has no overall electric charge. Okay, it is a Zwitter ion at neutral pH. So this is how it is represented. At low pH, there is the proton concentration is high, therefore this carboxyl group accepts a proton and the overall charge becomes positive. At intermediate pH, the overall charge is neutral, and at high pH, when there is low concentration of the hydrogen ions, then this NH2, NH3 plus will lose a proton and it will become NH2. That is the reason why the overall charge on the amino acid will become negative. So at high pH, the amino acid is negatively charged. At intermediate pH, there is no charge on amino acid, and at low pH, the amino acid is positively charged. Why? And this is how the amino acid is known as Zwitter ion. So that's all for this video. Thank you for listening.